to, and just kind of take a look at where the future is with that research. I'm planning for a review session on Wednesday. My thought was 4 to 6 o'clock. And then on Friday, we've got an exam. So that's what we've got coming up in class. In lab, after practical today, so Friday, I put this note up this morning, but I want you and your group to bring with you to lab on Friday your methodology that you've written up and I've given you comments back on. And then also the Guess My Hypothesis worksheet. And the reason why I want you to bring these is twofold. One, I meant to make copies of all these and then somehow I didn't save them on my computer. So I want to get a relook at everybody so that I can just make copies of them for the purposes of just kind of keeping a running track of where everybody's at. Second reason, some of the comments on there may not make sense. I want to use these as kind of a springboard for conversation on Friday. Each group will get 15 to 30 minutes roughly to meet with John and me, or just John, to talk about the progress of the project thus far, and then to kind of look ahead and think about, okay, so what do you still need to get ready before doing the experiments? First round of independent study days next week, Friday. Okay, so you'll be doing your first experiment next week on Friday. And we'll be making media for it on Wednesday. Sydney.
Did you say what time the review session will be on Wednesday?
My plan was 4 to 6.
Okay.
My plan was 4 to 6 because I didn't want to make it too late, but does that conflict with a lot of class? I don't really know what folks have on Wednesdays. I thought that was usually a lighter day. Except for Allie.
Yeah.
Okay. Well, I'm planning on doing 4 to 6, but as always, if you've got additional questions, if you want to meet with me one-on-one, please let me know, okay? Last thing for, anything else for lab? I think that's it right now for lab. Danica.
We don't have to turn our materials separately, it's already on our methodology that we turned in.
No, just bring it with you, because I want to just have a double look on Friday, just so that we know what are we going to have to make or prep in lab on Wednesday, and then what do John and I still need to order. Okay. I did forget, the reason why I want you there for full lab, regardless of when you're coming in to meet with John and I, we are going to do a little art therapy, so to speak, on Friday. So one of the yearly traditions with American Society for Microbiology is a project known as Agar Art. You basically get an agar plate, you get a palette of bacteria as your colors, and you paint. And then you see 24 hours, what does it look like? Actually, I have a contest for this each year with the winner getting a certain amount of money. So,
Wait, what?
Money.
Money, like if we submitted yours as one of those art projects, you could get money for it.
Okay.
I'm just saying. So we're gonna paint for a little bit. Use inoculating loops or cotton swabs. Yep, it's Friday. All right, so that's what you're doing during lab time. All right. Yeah, just an agar plate. That's your canvas. All right, and as Bob Ross would say, there's only happy mistakes, right? All right, no mistakes, only or whatever. I'm not a huge Bob Ross fan. Okay. So where I wanted to go today was to come back to bacteriophages, and in particular, what I really want to focus on are the potential benefits. When we think of viruses, we typically think of the downsides. We think of how they negatively affect our own health. So if you've had mono before caused by EBV, that really sucks. You're out for 3 weeks plus extra time because you're usually fatigued for a long period of time afterwards. Flu comes on like a freight train, about 2 days of absolute misery, and then another week of somewhat misery afterward. Okay? That's what we usually think of. What we are going to focus on is what are the potential benefits of viruses, and really the focus here are bacteriophages. So I asked you to take a look at this article here. And let's just start by summarizing. What happened in this article, or what does this article actually discuss? For those that read it, so I'm not making assumptions. Okay, so something with the heart. He had surgery, and then it gets infected with what? Pseudomonas aeruginosa. So, I'm going to stop a couple of times just to give some background info too. Pseudomonas aeruginosa, opportunistic pathogen. Okay, and what we mean by that is, in a healthy individual, most often Pseudomonas doesn't cause disease. But in the case of somebody who is immunocompromised, and that could be something like an open wound, it can cause disease, because usually somebody who is immunocompromised has a weakened immune system in some way. Pseudomonas infections are really severe in cystic fibrosis patients because they don't have the cilia, their cilia usually, and the mucus that they have in their trachea doesn't move correctly, and so what they get are infections within the lung of Pseudomonas aeruginosa. This one probably acquired in a hospital, so the term we would use is either hospital-acquired infection or nosocomial infection. Might be the other term that we use. All right, so Pseudomonas infection at the site of where surgery occurred. What happened after that? How many years did this, or how long did 3 to 4 years roughly. Consistent the whole time? No, why not? What did they do to treat it? So they used antibiotics. It would reduce the amount for a period of time. He'd be good for a month or two, and then rebound or recurrence of the infection. What's likely true is that the Pseudomonas probably never went away. Was that probably the antibiotics were good to kill the vast majority. And if you think about what we've talked about in terms of antibiotic selection before, we're probably over time selecting for strains of Pseudomonas that are more and more resistant to antibiotics than the previous ones. So treat, reduction in numbers stay low for a while, you get regrowth. The reason why Pseudomonas is such a concern when it comes to medical settings is because it is incredibly resistant to antibiotics. And it's not just because of mutations. It's not just because of efflux pumps. Actually, Pseudomonas has a couple of other features. One is that it can form biofilms. Anybody know what that term is? This is not one we've covered this semester, but is an important term to know in the medical setting. Anybody know what a biofilm is? So biofilm, in essence, is the best example I can give, plaque on your teeth. That's a biofilm. It is oftentimes, it can have the consistency of anything that's kind of like snotty to something that's more almost to the consistency of concrete in some cases. Biofilms are produced by bacteria, specific species of bacteria, and essentially help the bacteria to colonize or to stay within a location. So the plaque that's on your teeth, the bacteria that are producing that are basically living inside of, I kind of call it like apartment living for bacteria. You've got the structure, and then you have bacteria living within that three-dimensional structure. We also find that there's great diversity in here. So not only will you get one single species, but you might get 2 or 3 different species of bacteria, thus starting to create a community where some might be, is that Pseudomonas produces a slime layer, which is oftentimes called a mucoid layer. What is a slime layer? And this is a term that we briefly defined a long time ago, beginning of the semester.
Slime.
Yeah. The other term we may have used was capsule. Anybody recall what a capsule is? It's the glycocalyx. So glycocalyx takes on 2 forms, capsule or slime layer. Capsule is pretty much tightly bound to the cell. Slime layer can be washed away pretty easily. But the purpose of the 2 of them is very similar. Prevent phagocytosis improve colonization or attachment. The other thing that it does for Pseudomonas, makes it resistant to antibiotics. The antibiotics can't diffuse very well through that slime layer, and so it makes them a bit more resistant. So because of those characteristics, Pseudomonas itself, again, opportunistic pathogen, but what's so challenging about it is it's just really hard to treat, and you get these recurrent infections once the first one has come about, oftentimes. So he gets an infection of Pseudomonas aeruginosa. They treat it over and over again with different sets of antibiotics, and nothing seems to be working. Then what? Then what happens? Phage therapy, exactly. So not only use phage, but also use an antibiotic at the same time. And we're only going to focus on one of the reasons why they would do this. There are 2 or 3 other ones that they've done some follow-up studies. The group that proposed to do this has done some follow-up studies with. We're just going to focus on a small facet of this. Before we get there, all right, let's just go back to this question real quick. So what I asked you to do for homework was to take a look at this particular chart. And this is one we actually saw before. I did put a typo in there. The protein is not OMPM, it's OPRM. So just to note, because the worksheets that you have listed as OPRM. What is the general trend that we observe here? And these are minimal inhibitory concentrations or broth dilution experiments. What's the trend? Paige?
Like when you have the deleted OMR.
Yep, that gene.
Yeah, like when you have that deleted, you need a lot lower concentration of the antibiotic.
A lot lower concentration, is that what you said? Or did you say more? I wasn't listening. The peanut butter in my mouth was
Lower.
Okay. Less needed to kill, right? Jaden?
Having that gene deleted makes it more sensitive.
Yeah, that would be another way to say it too. It makes them more sensitive. In other words, this gene is required for resistance to antibiotics, right? And some people pointed out some information like, well, this is a efflux pump, and it actually is. Matt, you pointed out a really good reason why, actually I think we can say it's an efflux pump, and that was because if you look at these antibiotics up here, what did you say?
They're all different targets.
Yeah, they're all different targets. If this were a gene that was involved in translation, for example, we might expect that the only genes or the only antibiotics that we would see a difference in the minimal inhibitory concentration in the mutant versus the wild type would be the ones that target translation. But since these are across numerous categories of antibiotics, inhibitors of cell wall synthesis, inhibitors of, well, all 3 of those, I guess, inhibitor of translation, inhibitor of gyrase. Since there are different categories here, what this suggests is this is a gene that probably is involved in the efflux or import-export of these antibiotics. Okay? And that's exactly what this is. So it's an efflux pump. It also means it's an outer membrane protein. Okay? Think of the bacteriophage life cycle real quick. What is the first step of the bacteriophage life cycle? Or any viral life cycle. So we talked about 5 major steps. No, that's bacterial. It's a good guess, though. It binds to this gene right here, OPRF. So it binds to the efflux pump. And what we're going to take a look at over the next 30 minutes is what effect does that have then on the bacterial cell? Okay? So what I want to do to start, and I realized this morning that I forgot to draw a picture. There's 2 parts to this. I'm just going to give you the first one because I want you to make a couple of predictions that we'll take a look at the results of afterwards. But just to get started, I want you to think about, okay, so what are the major parts of a lytic life cycle for a bacteriophage? That's what's listed as A on the front page. And then B is what's a lysin. Anybody recall what a lysin is? I just figure we'll review this one right now. Is it? No? Oh. Jazz hands. What's lysozyme? Or what does lysozyme do? Breaks down the cell wall, destroys cells. Lysozyme is a specific type of lysin. So all lysins break down the cell wall, and in the case of a bacteriophage life cycle, help to release those bacteriophage. Okay, you might remember we saw that picture of like all the phage coming out of one end of a bacillus cell. And that leads to the formation then of these plaques that you see on this plate. Those plaques there are, again, single bacteriophage infects a cell, replicates, kills it, infects the ones around it, replicates, kills it, so on and so forth, so that what's left behind is just kind of like a hole in this lawn of bacteria. So I want you to start by describing the life cycle very briefly. And then on the back side of this first page, there's two other questions. I'm going to draw an image for the top one. And I want you to start to think about, okay, so what's going on during the life cycle of this virus? What do we expect to see then in terms of bacterial growth without or with this virus as well? Those are the first couple of steps here, okay? The first couple of paragraphs there are things that we've already talked about, so you can kind of just skip ahead to reading the problem, and we'll go from there. All right, I'll give you guys about 8 minutes, and then we'll talk. Go. Bless you. Feel free to work with somebody else too. You don't have to be quiet. What? No, I'm not mad at you. I'm just giving you a hard time. Or you can just kind of summarize. Occurring, none of the remaining steps can occur. So as Danica said, in this case here, we are essentially forcefully stopping that attachment step from even occurring. This is how we can, or this is how folks will identify receptors for bacteriophages, taking a library of one strain, each with a mutation in one of the different outer membrane proteins, and then test for, okay, so which ones are they no longer able to infect? The ones that they don't, that's our attachment receptor. Okay. let's take a look at the last one here, and then we'll take a look at the results together. And just to give you, who's Shannon Turner? They're the folks that have done these projects. If you're wondering, wait, who the hell is that? All right, so they're doing a spectrophotometric measurements of Pseudomonas aeruginosa, and I've given you some guidelines to get you started with this. The first is that it has a 1 hour log phase. What does that mean? Or oh shoot, that's supposed to say lag. 1 hour lag phase. My apologies. I'll give you a second to redraw it. So 1 hour lag means what? Takes an hour to get started. So something like that. And then we have what phase? Okay, so at that point we start to see increase in growth, and eventually that growth is going to peak out. Probably within this time frame we wouldn't necessarily see death phase. Okay. This also says that you actually have two cultures. You started them out the exact same. At 3 hours, one of them you add the phage, the other one you allow to keep growing. This is the one that we allow to keep growing. Okay, this is what would a typical growth curve for Pseudomonas would look like. What I want you to do is take a second and think about, what is this curve going to look like, or how is it going to change after 3 hours if we add bacteriophage? Okay. And draw it out for yourself. All right, now that you've had a minute to think about it, what would we expect to see? Add bacteriophage at 3 hours, and then what? Probably something like, did you take a look at the full case study? Okay. So maybe a short stationary as the virus is starting to replicate, and then once levels get high enough, death. That's exactly what we'd expect to see. What's driving the death? Well, it's the replication of the virus within those cells. Okay? Is what's driving that death. So what I'm going to hand out is the rest of this. Now, this graph looks a little bit different than the one that we just drew. And what I want you to do is take a minute or two and try to figure out, why does it look different? What you're going to see in this image is that there's a regrowth of the bacterial cells, and I want you to think about, well, what the heck has caused that to occur?
Yeah, and then the year before that, I was in 6 hours information, like I got some curves. Yeah, 6 hours. So it was like 200 kids.
All right, so the major thing that is different between what we've just drawn and what's here is at about 11 hours or so, we're actually getting regrowth. Why? What might allow for that regrowth to occur? Jaden?
Maybe kind of some mutation.
Yeah, exactly. Mutation of what?
The bacteria.
Yes, specifically.
The receptor.
Bingo. It's the receptor. Okay? So what we find oftentimes, and these we see even when myself and some other students are doing this in our lab, what we find over time is that you get good killing of those bacteria, but remember, these are populations, okay? And within those populations, there are going to be some that have different genetics. If we were to test the ones that have grown here by 16 hours and compare them to the ones that we started with, what we would find probably is that there's a higher frequency of mutations within OPRM here than there are in this initial population. Okay. What might that mutation do? One, it blocks the virus from being able to attach and even to infect those cells anymore. But what else? Given everything we've talked about, including the homework, what might that mutation do? And take a minute to talk it over with someone, and then we'll come back and talk about it. But think about, with everything that we've talked over the past 40 minutes, including the homework assignment, what might a mutation to OPRM do to Pseudomonas aeruginosa?
I know, that was a good one.
What are you thinking? I don't know, I didn't even hear.
After a while, the receptor will get resistant to change the new form of thing, and it won't.
Okay.
Because of the changes
That one really fun.
Okay.
due to mutation.
So the mutation's in the DNA, right? And that ultimately is going to affect the structure or the sequence of the receptor itself. And then what does that change in the receptor do?
Oh, yeah, I didn't really do that.
So that's one. That's one. And so the virus wouldn't be able to attack. That's why we see this grow, right? What else might be
I
affected?
think, but it
What is OPRM? Or what does this membrane protein do?
It could be.
Doesn't it like push the antibiotics out?
Good. So how might that
Yeah,
change
there's a big thing.
when
I thought one
that
was crazy, but it was pretty.
multi-drug resistance pump or that efflux pump gets mutated? Hypothesize is the mutation that's occurred here prevents the virus from being able to infect the bacteria anymore.
Yeah.
Yes. We could assume that from the results. Because the only ones that grow toward the end there are ones that have mutations in that OMP.
Aren't they all going to run to something?
Ah, they will, yeah. Yeah. So you're selecting for ones that have that particular mutation, right? What else might be the effect here? And for that we have to think more broadly again as to what is the function of OPRM? What is the function of this membrane protein? It's a pump to pump out antibiotics. So what might be the potential effects of a mutation on it? So, and these are likely missense mutations. They're not full deletions, but missense mutations where I'll just leave it at that. Missense mutations. What might be potential results of this? Say that, Sarah?
It can no longer pump out antibiotics.
So these cells would no longer be able to pump out antibiotics, making them more what? Sensitive or resistant? More sensitive. That's absolutely one. What else could be true?
Why antibiotics? Why would you try to infect?
What's that?
Why would you try to cure the virus with antibiotics?
So we're not curing the virus of antibiotics. We'll come back to this. We're going to come back to the concept of what the benefit of this is. But one possibility is the cells are more sensitive to antibiotics. The other is maybe this mutation actually makes those pumps better. Maybe it makes the cells more resistant to antibiotics. The third is maybe the mutation has very little effect on the function of the protein at all. Okay? So they took a look at that. They took a look at how do these cells respond to antibiotics? So here's that table, and this is on the next page. The strains that they are looking at here, just to orient you, PA01, PA14, these are kind of like prototypical lab strains. These two here were ones pulled out of a household kitchen sink. And then these bottom three here are actually clinical strains. One of these, I'm not mistaken, I think it's this, I think it's the PAPS was pulled out of the patient that was featured in that article. Okay. What I want you to do is, they've split this up into the OMKO1 sensitive, so the initial population of these cells, or ones that are resistant. So the ones that after that 16-hour of growth, we see the regrowth of those bacterial cells, those are what they're considering resistant to phage. What I want you to do is compare these two populations to each other. This is a broth dilution experiment, just like the one we looked at for homework. Describe the general trend, and I'm going to give you a minute or two to look through this real quick. So what is the trend in this data? What is the trend? In general, what do we observe for each of these various strains? How do the mix compare to each other? Sarah? Okay, so these that are resistant, those that are isolated toward the end of this growth curve, have smaller minimal inhibitory concentrations. Meaning what? Takes less antibiotic, therefore these are more sensitive to the antibiotic. So Zach, to kind of go back to your question of like, well, what's actually going on here? What they learned from this experiment was actually the specific region of this OPRM gene that the bacteriophage were attaching to. If the bacteria gained resistance to it, they actually, in this case with tetracycline, and they tested this with a number of different antibiotics, they found that the mutants were more sensitive to antibiotics. In other words, they have figured out a way to take a bacterium, Pseudomonas aeruginosa, that is typically really hard to treat with antibiotics, and now make it easier to treat with antibiotics. That's what they've done here. Really? What it probably, yes, but with also treating with phage at the same exact time. So the idea would be, use these in combination. There's a synergistic effect here. What this takes us to is a model known as evolutionary trade-off. Okay, so for example, in evolution we talk about selective pressures, or natural selection, the process by which certain genes get selected for. And we see that here. Our selective pressure on the Pseudomonas aeruginosa in this experiment were the bacteriophage. The only cells that were able to survive were ones that had a mutation in that OPRM gene. Okay? So that's advantageous. But there's a trade-off. What is the downside of those mutations that have accumulated in OPRM? They're sensitive to antibiotic. And so the idea here is to treat with both at the same time. And by treating with both at the same time, you're more likely to cure the infection than you would treating with one, just the antibiotic, or the other, just the phage, where you'll observe resistance to the phage within hours. That's the idea behind it. Does that help to clarify? That's exactly what they did with this patient. So they went to this guy who had had these recurrent infections for years, and Turner and Chan, I think, was the one that actually went to his hospital room and said, "We have this idea. We're wondering if you will try it out." Now, this has not gone through any sort of clinical trial. This was kind of just like a, let's see if it works sort of thing. It was a last resort type medication. The doctor approved, the doctor being the patient approved, and they treated him with antibiotics and phage at the same time, and within 24 hours started seeing improvements. His wound that had been open for 3 years finally healed and closed up. Okay? So that's the proof of principle behind this, is that this actually could work. Is it 100% effective? No, nothing is, especially when you take evolution into account as well with microorganisms. But let me give you one more example of where this is being used, or at least being considered right now. Pseudomonas is also a cause of chronic ear infections in children. And so there's a thought that instead of using antibiotics to treat those infections, because you can get recurrence, I know Jake had like 4 in 4 months, and that was a living hell. But what you can find is that if you treat with bacteriophage, that seems to cure the infections just as well as an antibiotic does. Okay? And that's where we're talking about, when we talk about the benefits, this is the potential benefit. If you want to see where medicine is going, I mean, I'm completely biased, but I think this is where it might be going, as we are running into more and more antibiotic-resistant strains of common bacteria that cause nosocomial infections or that are strongly pathogenic.